United States Department of Agriculture.
So hello, and thank you very much for inviting me to speak today here. I'll be discussing the approach that we used in Cardiff University for animal and animal tissue sharing and how that possibly could be implemented in other institutions. So I will start by just discussing briefly the current animal experimentations in the laboratories. So what we see, we're showing mouse here as an example, but what we see is there's two groups of animals. Firstly, there's some animals that go into experimentations. So for example, a group performs an experiment, there's a treatment on the animal, and then they collect specific tissues from that animals. And very often there will be very limited numbers of tissues collected, sometimes even one, and that goes to a single scientist quite frequently. The other group is the mice that are like mentioned by Dr. Freeman, which are basically wrong genotype, excess breeding, or just other sources of animals that are not needed for experimentations. And those are just being discarded. So considering that in America, there's about 700,000 animals reported by USDA to be used in 2022. If we include mice, rodents, fish, and birds, that rises to estimated 14 to 24 million. So considering those numbers, we might think that at least that many organs, tissues, cells, and animals that are wasted each year, despite the obvious demand for the tissues and animals. So we thought whether we can change that and change the practices that we currently see in the research. So what we're proposing is that animals, instead of being discarded, now being available in the platform, which is available within the institution, not between the institutions. And those animals can be made available to other researchers, and then they can be taken up by different group and the different project license and used for experimentations or tissue collections. Obviously, those mice would have to be there would have to be some legal clarifications because they might include mice that already or animals that underwent a procedure, and the new project license have to allow for the reuse of the animals. The second group of animals will be animals that have been used for tissue collection. And again, though the researcher who's performing the experiment can let others know ahead of the experiment what tissues they will be collecting and what procedure was done on animals, then other researchers can join and collect other tissues and organs from the same animal, and therefore reduce the need for breeding or purchase and still get the organs and tissues they require for the experimentations. So to verify if that could work, we performed a pilot study in Cardiff University. This was a limited participation study. We've used 10 active personal license holders from 2 research groups. We asked them to use mice only for this purpose. We asked participants to use the Excel document for announce any tissues or organs from mice that can be available for the experiments to others. And for the simplicity, we requested that mice destined for tissue collection only were used, not mice which have undergone any procedure. This is just to avoid any ethical issues. And what we've seen is within 19 months, 42% of total mice used by 2 research groups were made available for sharing. And that accounted to 256 mice. And out of that, 37% or 97 mice were shared between 2 and up to 6 users per mouse, which is quite incredible when 6 people collect tissues from the same animal. So consequently, we collected additional 205 tissues. This accounts for staggering 140 mice reduction that we calculated, which are possible in the presence of the approach. So if we extrapolate it, and if it's representative of UK mice usage, this would equate to a reduction in numbers of animals used in UK of estimated 70,000 mice a year. And if we calculate it accordingly to the numbers published in America, not published, but estimated numbers for the America, that would account for about 2.1 million animals. So it's quite significant reduction that can be achieved with the platform. So while the Excel sheet works for a little study like this, to implement this system on the institutional scale, we reached out to a company called Attune, which provides animal management software in different countries. They operate in over 20 countries and 140 institutions. including Cardiff University, and we asked them if they would be keen to implement that into their software. And they agreed, and we created an add-on for the software, which is a Ticket Lab, the basic software. The add-on is called FRIA Blackboard, and which basically allowed sharing and advertising animals and tissues on the Blackboard and for others to come and collect it for their own experimentations. So this was done in the collaboration with the Attune company, as I mentioned, University in Aachen, and us in Cardiff University. At the same time, in Bristol University, they developed a system which operates pretty much in the same way. It's called Internal Tissue Sharing Network, and they used Microsoft Teams, which is widely available in institutions worldwide. So it gives a massive potential for any institutions to implement it in their workflow. So just having a look on the FRIA Blackboard, this is how it looks. Basically, it's very simple interface. What the person sees when they check the FRIA Blackboard is the species of the animal that's allowed, the type of tissue or animal is an animal that's available, the quantity, and this description. So that includes strain, if there's any procedures being done on the animal, etc. Date of availability and expiration date. So these are different dates. So availability would be a period of time. So for example, when we set tasks to our animal facility workers for culling of excess animals, we have to give them a week to perform that task just because they're quite busy. So this would be that gap between the date of availability to expiration date will be the date of setting the task to the date of culling the animal or tissue collection or experiment if someone is just offering organs. So then there's information about the name of person who's offering animals, the email address, and the contact details to contact for collection. The FRIA Blackboard also allowed for people to request animals. So if I would need, for example, eyes, I can say that I'm looking for eyes from animal. I can specify the strain and so on. So it works both ways. So you can advertise, but you can also see what's available and use it in your time. So we decided to see how the efficiency work on the institutional scale. So we compared FRIA Blackboard and the internal tissue sharing network. And we observed that within a year, there was 336 animals and tissues available shared on FRIA Blackboard in Cardiff and 179 in Bristol on the Microsoft Team platform. And in relation to the annual usage of animals in those institutions, that equals to 1.2% for each institution. So basically, there was no difference in the performance of the sharing or reduction. And although 1.2 sounds very insignificant compared to over 30% that we achieved in a pilot study, what we have to remember is that in the pilot study, we're using only 2 groups that work on the same discipline. So they both immunology groups, and they labs are next to each other, so they will know each other. So we're expecting a bit higher efficiency of usage. Also 1.2%, it looks very low, but if we calculate it based on usage on the country, 1.2% reduction would be more than 33,000 animals in UK, over 100,000 in European Union, or over 168,000 in US. So there's a massive potential for the reduction, even when it's not working optimally as it is at the moment, and I'll come back to it now. All I want to mention is for this study and for further analysis I'm going to discuss now, we got funding from the Welsh Government, from the Innovation for All project, so thank you for our funders. Before I dive deeper into the change of effectiveness in reduction, I wanted to consider some other benefits of animal and tissue sharing. And this includes cost reduction. So if you don't have to buy or breed animals, obviously you're reducing your costs and cost to animal facilities as well. Increased productivity. So you have more access to animals and tissues, so you can test your reagents or do training or use animals for specific experiments. Increases in collaborations. We see that between the groups, and also knowledge exchange. So all those benefits are based on the feedback that we got from the users of the platforms, and they were validated. And one such example is this group that obtained 40 wild-type mice from the platform for their study. And they published it recently in scientific reports. Looking at the savings for that group, the average cost of the wild type animal in UK is between 20 and 50 pounds per animal. So 40 animals, it's saving in the area of 800 to 2,000 pounds for the group, for the paper, which is great, I would think. So is that initiative important? Is animal sharing and tissue sharing something that's needed in universities and wanted? So we asked that question on the conference for immunology and received the answers from 77 participants from 16 different institutions, including international universities. Participants of the survey included researchers at various career stage and those with and without personal license. So they would either work with animals or not currently for the research. And overwhelming majority said that yes, they would benefit from the access from tissues and animals. Some were unsure and some said they wouldn't benefit from it. So we also wondered, would they be keen to participate in the sharing? And we've found that over 90% were very keen to participate and advertise their animals if such platform existed in the institution. We wanted to investigate the reasons to get tissue or animals from the platforms, so we asked for that as well. And what we found is 3 main groups of reasons. Firstly, testing new protocols, reagents, protocol optimizations. So instead of using their own strains, which can be sometimes in low supply, that can be used, other animals or different strains can be used for those purposes, same as the training. And then those animals that can be used for these purposes include ex-breeders, for example, or breeders that have wrong genotype, as we've heard about them from gem colonies. Second group includes experimentation. So the reasons to get the animals will be experimentation. So there would have to be animals with the specific strains, which can be harder to come across on sharing platforms. However, they are very needed. Lastly, we have access to tissues I normally have no access to. So people indicated, especially those without the project license or personal license, they would benefit from access to animal tissues and animals to confirm the in vitro studies. So going back to reduction efficiency, we were wondering, after having implemented the platform in our university for some time, for a few months, we wanted to see what interactions people and users, so there will be researchers, had with the platform to date. And surprisingly, we had about 15% of people interacting with the platform in one way or the other. This included collection of animal tissues or animals, posting wanted animal tissue to request, or making animals and tissues available. 35% check what is available, which is quite encouraging. And there's another group, which, for example, some comments we got was that they responded to the request as well. But what was the most shocking is there was 53% of people who have not interacted with the platform at all. It was very disappointing because we put lots of effort in advertising of the platform. So reaching out to all the end users, we've done lots of presentations, emails, and use different approaches to reach to everyone to know this is available and how people can use it. So we wanted to know why they're not using it. And we identified 4 main reasons for or challenges that prevent optimal usage of the platform. So firstly, we have practicalities. So people say they don't know that the platform exists despite our efforts, like I explained. Additionally, there's people are not sure how platform works. So the guidelines are very important to know how the platform operates on the interface, I think, and also what should be the workflow of tissue and animal sharing within the laboratories and within the animal facilities. There's some legalities, as you may expect, like MTAs and terms of conditions. So there was quite a high percentage of people that were not sure about those. So they prefer not to share rather than break some MTAs. Ethics is a very massive topic when it comes to tissue sharing and animal sharing. I'll consider it a bit in more detail in a minute. But for example, the researchers were not sure if posting animals that undergone the procedure was ethical, which is not in all the cases. Also, researchers are not aware what is permitted under their license, so they do not use the platform to avoid any issues. Lastly, participation was an issue. some end users decided it's too much hassle, and I see I'm missing the text here. I'm trying to remember what I was saying here. Probably it was how to organize the collection efficiently. Other comments included, for example, that the background is not correct, that what they're looking for is not very commonly used, and also that the colonies are not big enough for sharing on the platform. That's why people do not participate. So focusing on the MTAs and legalities, we ask if researchers are aware of any legal tissue sharing restrictions on the animal strains they're working on. And interestingly, over 90% of total participants were unaware. The ones who said yes specified MTAs and brought attention to other sharing issues such as intervention or if the animal has been under procedure, which is more of an ethical concern rather than legal concern. That was a very important question to us because it clearly shows that end users of the platform have very little legal knowledge on the animal and license permissions. And if someone is unsure, that would stop them from using the platform or might use it and share animals unlawfully. So that has to be prevented or incorporated in the platform principles. So this, I just copied some terms and conditions from the Jackson Laboratory, and they clearly say that there can be some MTAs on the products and because they act as a central repository as well. And in addition to this, common terms and conditions, additional terms and conditions may apply, include separate rider label, use license, commercial agreements, and material transfer agreements. And in those cases, it's very difficult to know that unless you purchase the strain and you actually went out looking for that information. So this is something we would like to incorporate in the guidelines we're planning to prepare, and I will be describing that in a minute. So currently, we identify 4 main challenges for optimal performance of sharing, animal and tissue sharing on the institutional scale. Firstly, participation. So for example, how to organize experimental workflow and plan experiments so more people or researchers can join at the same time. Also, lack of recognition, whether if there was some recognition that would help with the grant applications. If you're sharing more animals, there's more and more questions being asked by the funders now about the strategies to implement 3Rs. If there was some level of recognition for those applications, that might help to encourage participation. We have practical aspects such as timings, room requirements, what has to be present in the animal facilities, how big they have to be, also access to facilities for those that do not have that access normally. And also a platform interface. It has to be obviously user-friendly to encourage participation and have to be straightforward. So for example, platforms that will be operating on a soft animal software, a management software like we have, for example, 3R Blackboard, it can be quite straightforward because you can just right-click and add your animal from your list of breeders or your colony list directly to 3R Blackboard. For Teams, for example, Microsoft Teams, you have to type that information. So that creates additional hassle and time really for people participating. Legalities, these are quite important. Material transfer agreement, as I mentioned, are one of the big considerations. Terms of conditions of animal providers, and also legalities imposed by Home Office or USDA in America, although this will be very little in that context. Lastly, ethics, and that is a very big topic. So the questions, for example, are tissue collection. Is it ethical to purchase animals for tissue collections only? Is it okay to cull animals for tissue collections? There's lots of institutions that use some level of tissue sharing at the moment. And talking to them in recent months, we've noticed there's lots of internal institution-based rules and policies, which are not always accurate to what is required by ethical organizations. So some institutions make it actually too complicated for the users to use the platform, and therefore will be decreasing participation. Animal reuse has to be considered, like I mentioned. For example, if the animal being used for some procedure, if it's moved to a different project license and they want to use it for procedure 2, they have to have the permission on their license to do that. So that has to be incorporated somehow into the platform to prevent, or guidance to prevent people doing it if they don't have the permissions. movement between the licenses has to be considered, and also whatever is imposed by the Home Office or USDAs on the ethics in that respect. So this is not very clear at the moment, and we're hoping to create some guidance in the future to cover this. So this is not an exhausting list of things that has to be considered for tissue sharing, but that's something we're currently working on. So as I said, we're going to apply for funding. We're currently writing an application for Partnership and Impact Award from a National Center for Reduction, Refinement, Replacement of Animals in Research. So the title of our application is Share It, and we would like to create guidelines for global implementation of animal and animal tissue sharing approach. So the aims of the project are to evaluate the reduction effectiveness in international institutions. So we want to use different platforms, including those that are using animal management software, so they're more advanced, and also those like Microsoft Teams that are person-run basically, or Excel. We want to establish comprehensive guidance for best practice, limitations, principles for implementations, the approach internationally on any platform. So we'll be considering legalities. Again, we'll be talking to companies like Jackson's, like I mentioned, also MRC and other repositories for animal strains, to understand the terms and conditions and how those can be adapted for the purpose of tissue sharing, if they can be adapted, and also how limitations can be implemented into different platforms to prevent unlawful sharing of animals. Lastly, we'd like to define system ethical criteria and system review criteria for institutional ethic boards for successful implementation in the establishment. So for that reason, we set up collaboration with ethical organizations in 3 countries at the moment. We would like to run interviews and workshops to collect the information, what's actually currently ethically available, are there any guidance, and just make it clear and write the recommendations how establishments can review this process on the university level and drive implementation of that in the system. So we're currently seeking project collaborators. So if you're currently using any tissue sharing approaches, we will be very interested to hear from you. We'd like to learn from you. Additionally, if there's anyone who would like to collaborate on the project and is willing to contribute to our ethical conversations or legal conversations, we'll be very interested to talk to you. And also institutions that are willing to introduce the platform in the institution on Microsoft Teams, or are current users or plan to use Ticken Lab, and would like to consider FreeR Blackboard add-on, we would like to hear from you as well. So if you would like to be officially involved in the project, I would ask everyone to email very soon, because the deadline for submission is the 10th of September already, which is next week on Tuesday. However, we'll be very interested to hear from anyone after that date as well, so we can take it on board and hopefully work after the deadline as well. So in summary, I hope I convinced you that animal tissue sharing has the potential to have a significant impact on animal reduction in research. And it is in principle easy, but there's some significant challenges that has to be resolved before it's optimally used globally. There are numerous benefits to sharing other than reduction. Researchers and animal facilities are keen to participate, and active participation can take time to develop. And active participation requires changes in the researchers' attitudes, experimental planning, as well as development of clear guidance. There are 4 main challenges that currently prevent optimal tissue sharing on the institutional scale, and detailed guidance for individual region and countries have to be developed, and those challenges have to be addressed. So I will finish here. I would like to thank everyone who's been involved in that project so far, from Cardiff University, Attune Software, specifically Andreas, University of Aachen, and Bristol University. And if you'd like to contact us, please contact on FreeR Blackboard at Cardiff email address, or you can contact me directly on my work email here. Lastly, I'd like to thank our fine funders, BBSRC, Wellcome, and the Welsh Government. Thank you.
USDA is an equal opportunity provider, employer, and lender.